Next up, I would like to welcome Ting Xu here up to the microphone. Ting is from the Child Mind Institute in New York and has been doing fantastic work developing approaches to align imaging data between humans and non-human primates. So welcome, Ting.
Hi, everyone. My name is Ting Xu from Child Mind Institute. Before I start, can you hear me now? Oh, much better. Thank you. Before I start, I would like to thank Alex and James organizing this symposium. And I have nothing to disclose. Okay. So look at these different brains. So different species have formed their own distinct brain. Then this raises a question. Throughout evolution, how have different species developed their present configurations? And I believe that just before we learned a lot about the wiring principles across species, and later after, and I believe we can also learn a lot about the evolution rule in morphology. Here, I want to focus on the functional organization first. And as you can see on the left panel, on the left panel, we can find the functional connectivity profile very similar across species, even in the high-order regions. The example here is the middle frontal lobe. And in the second panel, you can see that we can also define the functional networks across different species, marmoset, macaque, and humans. And on the third panel, you can see that the brain activity during the nature vision, which is the same movie played to the human and the macaque, also have similar regions activate in the same way. And the last panel shows the dynamic coordination across different species, rodents, a macaque, and human share very similar functional dynamics. So if we look at this evolution tree, we can easily find corresponding common areas across different species. And combined with the evidence showed before about the functional organization across different species, it's natural to think that in addition to common area, we probably would find common access across species as well. So Daniel Margulis has this nice work using fMRI to delineate these functional gradients, capture this functional organization, and you can see in this low-dimensional space, it has very similar functional organization with macaque as well. And the first dimension delineates the hierarchy axis from the early visual and sensory model to the deformed network. And the second gradients capture the modality gradients from the visual to the sensory motor cortex. So we can use this to align species. This is the work that's been done in 2020 that's basically used these homologous regions as landmarks to extract the common axis between human and macaque. Specifically, we define those homologous regions as landmarks and calculate the functional connectivity from each vertex to those landmarks, and then build this human to macaque similarity matrix, put them into the diagonal, off-diagonal, and to build this joint similarity matrix, and applying diffusion embedding to extract the matched component between two species. And the lower panel, you can see that this is the component, match the components back to the surface. And the advantage of this method is that we can find those components cover the whole brain, well, the whole cortex, and it can also delineate highly similar regions regardless of the divergence of the anatomical locations. For example, the macaque visual cortex usually extended to the lateral occipital lobe, but for human, it's mainly in the medial side. And the empty region in human is at the occipital lobe, but for macaque, it's in the temporal sulcus. And using all of these functional components, only showed 3, but we have more. And using these functional components, we can build these features to put it into the spherical matching Well, recently I just learned that that might not be the best, but let's see, this is previously done. And we can use these features to build the surface matching. And on the lower panel, you can see this is a validation that after we have the transformation between two species, we can map the T1, T2 ratio map from human back to the macaque surface and compare to the T1, T2 ratio map directly extracted from the macaque, and they are highly correlated. And apply the similar technique, we can also build the macaque-marmoset transformation. As you can see that in this low-dimensional space, macaque and marmoset matched components are aligned very well in this space. And if we project the component back to the surface, similar thing, we can see that they are also matched from the visual to sensory modal for the first dimension, and the second dimension from the VMPFC to the sensory modal. And the third dimension is more broader deformed network to the visual and sensory modal cortex. And similar way that we can use these features to do the surface matching and use macaque as a middle species, we can align marmosets and the human together. We can also do it for macaque and extend it to the rodents. Here's an example for mouse that you can see the similar components capture the visual to the sensory modal, including the auditory with visual as well. And the second component is from the visual to the frontal lobe. And in the low dimensional space, these two species also aligned well. But at this moment, it's very challenging to align the surface together. But using these functional features, we can get the instant cross-species and within-species similarity. As you can see that this demo just show wherever I click in one species, I can get the within and cross-species similarity very easily. Of course, the mouse surface has been magnified 100 times. It's not that big. And once we build this vertex-to-vertex similarity matrix cross-species, and then we can see, okay, let's measure for each vertex, what is the local maximum from one species to another. So take the first one, macaque to human, as an example. So every vertex in human, the number here represents to what extent that I can represent this human functional connectome by using the macaque functional connectome in the similar regions. So the warmer color represents the highly convergent, basically homology regions, and the cold color represents the divergent regions. And as you can see, for example, the macaque to human and marmoset to macaque, all of this homology map shows a high homology index in the low order regions, for example, the visual, auditory, sensory modal regions, and the high order regions like deformed network and frontal network have the lower homology index. And we can also compare, for example, this macaque to marmoset and human to marmoset. So consider macaque and marmosets are closer species in evolution, so naturally we can have higher homology for macaque to marmosets, much higher than the human to marmosets. And what I want to highlight here is what I find interesting. If you look at the blue region, which is the divergent region in this human to marmosets map, that I think the blue regions suggest that where in the evolution, that's where our surface expansion to start. So I know that marmoset is not the common ancestor of all three species, but let's see, presumably, marmoset is the species that more close to the common ancestor. So I think this would provide a clue that's the evolution of the brain folds to start from, let's see, the lissencephalic animals to geroencephalic animals. Then the next question I want to ask is, which functional area can be characterized across species? So in human literatures, that we can look at the functional profile to identify the borders. For example, here, the supramarginal gyrus and angular gyrus are neighboring regions, but the functional profile, the functional connectivity to these two regions are very different. So we know there must be a boundary between these two regions. So we can go step by step and to check the functional profile. As long as we are across the border, then I expect there are dramatic change of the functional profile. So this is a way that we identify the borders in human. And of course, this is a very nice map using all different kind of features to identify the borders. Now imagine we do this in human, but imagine right now we have the macaque human similarity matrix. So instead of using the functional features within species, let's replace this human to human similarity matrix with the human to macaque similarity matrix. In another way, that we want to define the borders based on the common features between two species, here the human and macaque. Now here's the result. Let me put the results, this boundary, overlap with YAL7 network, just the visual and the sensory modal network. As you can see, the border align with this, visual and the sensory networks border very well. And if we can zoom in a little bit to see the visual cortex, and you can also see it aligned with the V1 border very well. And even it can differentiate the ventral and the dorsal, this border, very well. However, if we look at the high order networks, it turns out only the border that differentiate high order networks and the low order networks that can be overlapped with the cross-species boundary well. For the borders like within those high order networks that we cannot differentiate very well. So this suggests that the high order networks may not be clearly segregated during the early human evolution. Now before we're doing the functional boundaries using human macaque similarity on human surface, how about we do it another way around? So basically define the functional borders on macaque surface using the cross-species similarity. And this is the result. As you can see, most of them are basically aligned with the south side gyri. And I give the 4 examples here, pre- and post-central gyri, and the middle frontal sulcus, cingulate sulcus, and the pre-paracentral sulcus. And I also highlight the reference here that you can see all of this seems to be aligned with the conserved sulcus and gyri very well. So this suggests that maybe the functional boundaries kind of rooted in evolution. Now I want to bring the comparison between species to the next level, comparing the brain developments between two species. So thanks to the data sharing, we have over 20 research sites that shared over 1,000 data sets to be able to do this work. But the first question you probably wonder is why this matters. So all the animal models requires accurate matching. We already have the matching of the brain in 3D space, but we want to add the 4th dimension, which is the time. Because we not only want to match the brain, but we also want to identify the matched developmental stage for study the brain development in human and agent, maybe the relevant psychiatric or neurological disorders. But beyond that, I want to introduce this Newtonian theory. What is Newtonian? You can see this is a chimpanzee baby, and this is a human baby. And the heads of these two babies are very similar. And when we're getting older, the difference, then it seems like human adults just stay as it is. So it requires, if we put this human development into the chimpanzee development scale, we'll see that the human have a prolonged early development, so make us have more time to mature. Now let's look at the data. Here is the very nice effort that Richard and Jacob and others make all the collaborative work I think they collected over 100,000 data sets that's made these human charts. As you can see, it delineates the development trajectory across the human lifespan. And here is the macaque chart we built. The first row is the raw data, and the second row is the trajectory we built, and third row is the trajectory for each site to see that if our data is biased for any sites. I will show the second row together with human data later, but basically we see the gray matter, wet matter, subcortical gray matter follows a very similar pattern between two species, but have time difference. Here I just want to highlight the fourth row, which is a growth rate. And I have orange color. Basically, this orange shade covers the stage before birth. As you can see, for macaque, the peak of the growth rate is always before birth. And for human, it's always after birth for the gray matter, wet matters. So this means that for development speed, human already slowed down even at the infant stage. And human baby in the primate lineage is considered as a preterm baby. Now, one can put these two together to have these global trajectories for two species. And a little bit background about macaque first. A macaque lifespan is about 25 to 30 years, and the developmental speed is 3 to 4 faster than human. So you will see the age is not fully aligned. We're trying to align them using these speed differences. However, even we consider those time differences, you can clearly see that macaque shifted leftwards, which means that macaque has faster maturation than the human, or another way around, human has prolonged development. And for the gray matter, wet matter, or especially for the cortical stuff, for example, the volume, area, and the thickness, and you can see in macaque, pretty much matured before childhood or even toddler. And for human, it's like a late childhood and even extended to adolescent or early adult. And then let's see the regional plot. So this is the trajectory that you can clearly see for the human, it shifted rightwards and it goes up and then goes down. And for macaque, when it goes down, it's not as obvious as human. And there's only one region that remains similar size, which is modal regions. Which makes sense. This is a function that's very essential to monkeys and for their survive. And on the right is we go step by step, make the decoding for the step-by-step growth map, which means in macaque, we have the window size of 1 month, and for human, it's 3 months. We decode the step-by-step growth map using neuroscience, and then we can identify the critical age for two species. What is the function that develop for this stage. So similarly, I don't know if you can see it clearly, the modal function is the only function that macaque has a longer developmental stage than the human. And for all the other functions, human has always a delayed and a longer maturation time. And if we compare across different functions, then you can see the unimodal, the multisensory functions in human that matured early, and all the high order, for example, emotion and verbal language, and those high order association, cognition, those high order functions still develop later. And then I want to bring you this website that we built. You can just use your phone to check it. It's called interspecies map, no space, .childmind.org. This is still ongoing work, but if you log in this website, you'll see that this is interactive. This is an interactive website. You can double-click on this surface, and then it will show you the within species similarity or the region that you click on, but on another species. And below that, I can find the cross-species, the within species lifespan trajectory, and you can also select if you want to show the volume, thickness, or the area. And on the top, they also provide you the neural correlate decoding of the region that you selected. So to conclude, I want to thank the team and all my collaborators and all the non-human primates researchers who generally share that data to make this work possible. And I also like to thank my funding source and the ChildMind Institute. And this work, Sam did all the cool animations, and he presents a poster, which is yesterday and the day before yesterday. But you can find him here. And so we have another poster about the functional dynamics in childhood and early adulthood, present by Kyung-Sung tomorrow. And thank you for your attention.
Thanks very much, Ting. That was absolutely lovely. We've got time for one question, so I might just ask the most upvoted question. Did intersubject variability, especially for humans, affect the alignment results across species?
I would say yes, but not so much, because the inter-subject variability compared to the cross-species variability is relatively small. So, yeah.
Thank you. Thanks very much, Ting.